let's see how this goes. Hopefully it works. All right, so this session was supposed to be what we were going to do on Wednesday. I was dealing with some stuff at home, so I apologize. I'm actually still dealing with it, but I'm here today to give you guys the last bit of review that you guys need for the exam. So I wasn't able to finish this on Monday, so I'm going to finish this, which was the very last question on the Chapter 5 SI. Then I'm going to do a little bit of Chapter 6. Chapter 6 actually probably is the least, well, I wouldn't say least important, but it's probably the easiest one to study for the exam, and I'll let you guys know why that is. It's practically a pure memorization chapter. But this is one of those in Chapter 5 that's a little bit weird. You're not going to find anything like this in Chapter 5, but the information in Chapter 5 and 4 you can use to solve this. So let me break it down. First is this thing here with a bunch of lines and stuff. Let me explain what that is. I'm going to flip over to the document camera. So, in the lab, in molecular biology, biochemistry, one of the most common things we do is we measure what type of proteins are in something that we're measuring, or what type of amino acids are in something we're measuring. One way to do that is to do what's called gel electrophoresis. Some of you may know what this is, some may not. What gel electrophoresis is a procedure to separate proteins, to separate amino acids, and you can even do it to separate mixtures of DNA and RNA. The way it works is that, imagine this square represents like a cask, and it's standing vertically. Okay, this is the top, and this is the bottom of that. Okay. The way it works is that in the lab I have this mixture of protein or mixture of amino acid, and I put it in this little hole up here. Okay, this is where the mixture goes. And I might have several different mixtures I want to try separating. So I have several different, these are called wells. I have several different places to put them. Okay. What then happens is, and let me make sure I get the charges over here, is that I input an electrical current here, where that's the positive charged end, whoops, that's the positive charged end and that's the negatively charged end, okay? And this electrical current, what it will do is it will separate your mixture into, the correct terminology is bands. Each line is a band representing a particular protein or amino acid. You can kind of guess which lines represent what types of amino acids based on where they are in the gel. A line here, closer to the positive end, this line probably represents a negatively charged amino acid or negatively charged protein. Some of you may know in your studies, remember those different categories of amino acids? What category of amino acid is likely to be represented by that line here? Acidic, right? If you guys don't know, acidic amino acids are generally negatively charged at neutral pHs. So any lines that show up near the top of a gel, and they will have the plus and minus labeled on there for you to remind you, any lines at the top are going to represent acidic amino acids. You can use the same type of logic to guess that the lines near the bottom here are going to represent what? Basic amino acids, which are generally positively charged at neutral pH. So to read a gel, that is the first thing you need to try to orient yourselves with is, okay, what does this line possibly mean? Acidic amino acid. This one must mean a basic amino acid. This one here must represent either polar or non-polar amino acid. Depending on the context of the question, they'll give you enough information to where you can figure out what amino acid they're talking about. And we'll see that in a second. That's the first thing you need to try to do is to orient yourselves like this. So that's what you should look at in column by column. This next column represents a different mixture, this second column. It's going to have maybe some lines that are the same. This next mixture looks like contained the same acidic amino acid as the first mixture and the same basic amino acid as the first mixture, but it doesn't have anything here, all right? So it's missing whatever amino acid that one was representing. The third sample might just look like that, okay? So each column represents a different sample, a different mixture. Other tips here. Okay, so I'll start with that. Another factor here, and actually I'll use the problem as an example for this. No, one step before that. Anyone have any questions on how to interpret a gel so far? I'll give you guys an example in a second. Yes?
Will the nodes always be placed like the positives on top of it?
Yes. If there is nothing labeled, you can assume always the top is positive and the bottom is negative, but it will be labeled on the exam.
What if the pH wasn't stubborn?
So also in the lab, when you do this, it is always neutral pH. So yeah, you won't have to worry about that. And the problem won't even bother to tell you that. You just go in and assume that that's what happened. Mm-hmm. Yes?
Can the problem ever say something about the gel being polar or non-polar?
About the gel being polar or non-polar? No, but you're talking about this line right here, right? They'll tell you what amino acids were in the mixture. They just won't tell you which one is which. So you can guess, oh, this one was the acidic one that they told us about. This one was the polar/non-polar one, and this one was the basic one. Okay. All right, so let's look at the example. Flip back to that. Okay. All right. So, you will be given one problem that looks like this, where they will talk about some disease. It'll be a long word problem style thing, but I'm going to break it down for you to the easy parts. It'll be a word problem describing some disease, and they will tell you in our example here, let's see, this disease has a mutation in a gene that produces a crucial protein that has alanine. So the mutation produces alanine. The gene produces alanine instead of the correct one, which is aspartic acid. So a healthy person, for whatever this mysterious gene is or whatever disease is, the healthy person would produce aspartic acid, and the disease carrying person would produce alanine. Then you'd be given a gel with patient A, patient B, patient C. And then they'll ask which patient has the disease. One of three possible questions, I should say. Which patient has the disease? Which patient is normal and healthy? And the third possible option is which patient is a carrier of the gene. For those who haven't taken genetics yet, or it's been a while since you've learned the genetics part of biology, remember that we have 2 copies of every gene. So it is possible to be a carrier of this mutation but not have the disease. You are a carrier when one copy of that gene has the mutation, but the other one is completely normal. So this heterozygous carrier of the mutated gene, whatever his gel is, and we're going to look at it in a second, that person must produce both aspartic acid and alanine for this gene, because he has one gene that is fine, so it should still be producing aspartic acid, but the other one is producing alanine. The one with the disease, both copies of the gene have to be broken. Both copies of the gene must be producing alanine, so there shouldn't be any aspartic acid in the person with the disease. So let's try to break this down. All right. Which one, after all that, which one do you think is the one with the disease, the one producing alanine here? B. All right, so here's why. That is correct, and here's why. Let's figure out, the first thing you should figure out is what does each line represent? What is it likely to represent? Well, we just went over that in the gel electrophoresis, that this line right here, which is closer to the positive end of the gel, this line must represent a negatively charged amino acid, which we just concluded was an acidic type amino acid. So the lines at the top must represent someone producing aspartic acid. So let me add that here. Must represent someone producing aspartic acid. The one at the bottom, we know, it may not be talked about in the word problem, but what type of amino acid must be represented by the bottom line? A basic amino acid. Well, the problem didn't talk about any basic amino acids, whatever. Okay? And now how about, raise your hand if you haven't been signed in yet, by the way. Okay, so it's just the middle 2 rows. So therefore, the middle lane, the middle lines, I should say, must represent someone who's producing alanine, because that's the only amino acid that's left here. Okay? All right, well, that means patient B is producing alanine, and only alanine. Doesn't matter what the basic amino acid here is. This patient is producing alanine, so this patient B must be the one with the disease. Okay? And again, it doesn't matter if you've never heard of the disease that you'll see on the exam. You can still answer this question. There will be a lot of times like that in the semester where they'll give you some crazy looking stuff. But remember that technically they will only ask you about stuff that you that you learned in the notes in class. Of course you haven't heard about a fake disease called bonitis, you know? You have heard about acidic amino acids and basic amino acids and nonpolar amino acids. So that's what they're technically testing you on. All right. Patient C, we're going to ask it the other way around. Patient C, what type of patient do you think this is? Is this a carrier or is this a normal healthy person? Normal healthy person, because they're only producing aspartic acid. Okay? Therefore, process of elimination, patient A must be what? The heterozygous, the carrier of this disease. All right? Because they are producing both aspartic acid and alanine. All right? And that does happen a lot. In terms of genetic diseases, most genetic diseases only happen when you have the mutation in both copies of your chromosomes, of that gene. Okay? Any questions on how to interpret this? All right. Okay, so now we'll move to chapter 6, and this is what I wanted to go over Wednesday. So forgive me for not being here. So let's see. Make sure I included everything I wanted to. All right. So yeah, chapter 6, and it is sort of like the 6A, 6B in the PowerPoints. Isn't that bad. Basically, it's memorization. This is the most important stuff you need for the exam, and this is sort of like the review part for chapter 6 as well. Is in chapter 6 they talk about the different levels of protein structure. So chapter 4 and 5 you learned about amino acid sequences, which are all those amino acids put together via peptide bonds. And now you learn about actual proteins that are made from those amino acid sequences. You should know, and there may be slightly more detail in the notes, that the primary protein structure is your amino acid sequence. It's just your amino acids connected to each other via a peptide bond. But that has no function to it. In order to have a functional protein, that amino acid sequence needs to fold in on itself, wrap around itself to form a three-dimensional structure. The first folding and wrapping around forms what's called a secondary protein structure, of which you learn two categories in this class. What's the first one that you learned for secondary structure? Alpha helix is exactly right. That is one of your two possible secondary structures you need to know for the exam. And what's the other one? Beta sheets, right? Alpha helix and beta sheets. And somewhere in the notes there's a slide or two about alpha helices, and they tell you like a bunch of cool, interesting facts about it. Those cool, interesting facts may show up as questions or multiple choice answers on the exam, such as, I don't have it here obviously, alpha helices are right-handed helices, right? I'm pretty sure there's a slide somewhere that talks about that. And alpha helices, and I don't remember the exact number, have 3.6 residues per turn of the helix or something like that. You will find the slide in your notes that will tell you that. And there's other facts on those couple slides about alpha helices that you need to know for the exam. Don't worry about anything like 90 degrees, 10.5 degrees, whatever. That won't be asked on this year's exam. So in fact, this thing right here is actually a little out of date. But just know the facts, like the textbook, well, I say textbook, but the PowerPoint facts of these structures. So alpha helix, beta sheets. There's one kind of problem-solving thing about alpha helices you should know, and that's actually this type of question here. Let me bold it real quick. It's this question here. Make sure this is still recording. Yes. Okay. Raise your hand if you haven't signed in yet. Just a no. Okay. Can you pass this all the way to the back real quick? All right. You may be given a sequence of amino acids, and you will be told that it forms an alpha helix. And when you go review your alpha helix notes, you'll understand that alpha helix is held together by hydrogen bonds. Okay. Those hydrogen bonds always form between. In your notes, they call it the N and N+4 rule. So look that up in the notes. All that basically means is that from this amino acid, which we'll say N equals 1, it must form a hydrogen bond with an amino acid 4 hops away. So 1, 2, 3, 4. So, the answer therefore for which amino acid forms a hydrogen bond with aspartic acid, well, just hop 4 times to the right, and the answer is leucine will form a hydrogen bond with aspartic acid. Okay, that is the only kind of problem-solving question with alpha helices that you'll have to deal with on this exam. Okay, they may ask methionine, so you know, just for the heck of it, which amino acid must hydrogen bond with methionine? It is alanine, right? If you want to see what this looks like, there's a slide somewhere in your notes that show that these hydrogen bonds are drawn, and this may help you visualize this. So an alpha helix is helical shaped, obviously, but how does it hold this helical shaped form? Well, this amino acid is going to have a hydrogen bond with this amino acid, which is, if this is N, this is N plus 4 hops away. So that's where the N plus 4 comes around, is that every fourth amino acid happens to line up perfectly over the 4 amino acids before it, okay? So that's what the N plus 4 rule is, that there's a hydrogen bond connecting these two things together. All right, let me go back to the worksheet real quick. Okay, this is still going. All right. Let's see. Yeah, don't worry about this thing. I'll get rid of it on the worksheet later. Yeah, no, the identifying characteristics of alpha helices and beta sheets. Oh, and this special fact here, of your 20 amino acids, one of them does not like to be a part of helices. Which amino acid is that? Anyone know? It is proline. Make sure you memorize that. That's somewhere in your notes. Yes?
For number 3, like if he asks us like for like alanine or something like that, if there's no hops to the right, could he ask us something like that?
No, he would have to give you enough that the answer is there somewhere, yeah.
So proline is going to be part of any helix?
Right, proline is, the nickname for it is that proline is a helix breaker. It does not like to be a component of any helices. So if you have a sequence, an amino acid sequence with a lot of prolines in it, it is not going to form an alpha helix, okay? So that is the amino acid that prevents the formation of helices. And if you go look at the structure of proline, it has a unique feature that none of the other 19 amino acids have, right? It is the only one whose side chain happens to wrap around and connect with the backbone of proline. No other amino acid does that. All the other amino acids, the side chain just is on its own. It does not wrap back around and connect to the backbone. Again, for the exam, you do need to know your 20 amino acids, name, structure, 3-letter code, and in my opinion, the most important thing is what chemical classification you would call that amino acid. Is it acidic, basic, polar, or nonpolar? So make sure you know those things about every amino acid. Let's see, the other features. Don't worry about steric constraints, or that. All right, so this stuff I believe is in 6B. So after you learn about alpha helices and beta sheets, which are your secondary structures for proteins, when you start putting alpha helices and beta sheets together, random assortments of alpha helices and beta sheets together, your protein is going to fold a little bit more and it becomes a tertiary structure of a protein, which is a 3-dimensional structure of a protein. The tertiary structure of the protein is your first possible level where that protein is functional, okay? So once your protein becomes a tertiary structure, a lot of the time it can actually do whatever function it was designed to do. Maybe it was an enzyme. It's going to go catalyze reactions. Maybe it was a structural protein. It's going to go to whatever area of the body it needs to go to. So that is the tertiary structures when you combine a bunch of alpha helices and beta sheets together. For the exam, there are 3 tertiary level proteins you need to know about and the details about them, and that is alpha keratin, beta keratin, and collagen, okay? So what I mean by details is similar with alpha helices and beta sheets. You go look at the PowerPoint notes, there's like 1 or 2 slides on each of these things, and they have bullet point facts about them. So in fact, for those who maybe have been studying that, we may be able to sort some of these things, some of these facts here into their respective categories here. So I'm going to see how much you guys know. One of these things is a component of silk, of smooth silk. Anyone happen to know which one? It is beta keratin, okay? And to go in like the biochemical reason why that is, beta keratin is just made of a bunch of beta sheets stacked on top of each other. Beta sheets are very flat structures, so you stack a bunch of them together, you have a very smooth feel to it. So silk fibers are made of beta keratin. So just go ahead and put that here. All right. Which one of these things are you going to find in hair, fingernails, claws, horns? That would be alpha keratin. Okay, good. Yeah, it's just memorization. Oops, this is going to be annoying. Okay. Yeah, it's just memorization, so you need to know alpha keratin is found there. And therefore, process of elimination, which one of these things is a connective tissue that you find in cartilage, tendons, bones, and teeth? It must be collagen. Okay. All right, now for the slightly more difficult stuff. Each of these has a pattern in an amino acid sequence that is unique for that structure. We'll start with this one. One of these things down here, when you read its amino acid sequence, we use the phrase 7 residue repeat. What that means is that you write down 7 amino acids, and those exact same amino acids are repeated over and over and over again. Anyone know which of these is a description of that sequence? That happens to be alpha keratin, okay? Be very careful. That is not alpha helix. This is alpha keratin. Alpha keratin, when you look at a sequence, if you're given 3 sequences, and we'll do that practice question in a little bit. If you're given 3 possible sequences, the one that has a 7 residue repeat will be alpha keratin. Okay, and all that means is 7 amino acids repeated over and over. There's this extra fact attached to it, and you can memorize it if you want. Also, the most important thing is the 7 residue repeat part. The extra fact is that the first and fourth amino acids are nonpolar, usually, for alpha keratin. All right, this next one here, there's a lot of info here, so it should be very easy to identify which one of these fits that because it's a very unique fact. This structure, a third of it is composed of glycine. This structure actually contains proline and unusual modified amino acids such as hydroxyproline, number 4, 3, and hydroxylysine, number 5. Which of these structures, anyone happen to know, happens to fit that profile? Collagen. Okay, collagen, when you go look at your notes, there's a phrase, annoyingly, where they say it's a triple helix. But didn't I just say that proline is a helix breaker, right? The triple helix, don't spend too much time trying to figure out what that means. It's just the overall shape of the thing. It's not a true helix, okay? So what I said earlier about prolines being helix breakers is still true. Collagen just has this weird 3-dimensional structure that happens to contain prolines, and it looks like a helix, but it isn't, all right? It's a little bit weird. Bottom line, the fact you need to know for the exam is this sequence information is matched to collagen, okay? All right. Well, process of elimination, this one, an alternating sequence of glycine, alanine, glycine, alanine, glycine, alanine, or glycine, serine, glycine, serine, glycine, serine, must very clearly be beta keratin. The smooth shape, the very flat shape that beta keratin has is due to this alternating pattern, in fact. Glycine is your smallest amino acid, and alanine is your second smallest amino acid. They contain the fewest. Well, glycine obviously is a side chain of hydrogen, and alanine is a side chain of methyl group. So it's a very, very small amino acids are used in the construction of beta keratin. So you make these sheets that can fit very close together. All right. Tropocollagen subunits, triple helix. I gave this away. What is that one? It is collagen. Okay. Made of anti-parallel beta sheets. Also gave this away. Yep, this is beta keratin. Made of alpha helices. So notice the names alpha keratin must be made of alpha helices. Beta keratin must be made of beta sheets. This one again. This one you actually, from what I can tell, you don't need to know for this exam, but because it's on here, I'll give it to you anyway. One of these has a wider pitch compared to alpha helix. What that means is that its helical looking shape is just bigger. That's it. So the answer must be collagen because the other two things don't form helical shapes. All right. Lastly, which one of these has a bunch of glycines on one side of the sheet and has either alanines or serines on the other side of the sheet? If we look at a picture of beta carotene, it'll look exactly like that, where all the glycines in the alternating pattern are on one side and everything else is on the other side. All right. Last bit of sort of problem-solving type information from chapter 6 is this. Which of the following sequences will form alpha keratin, which one will form beta keratin, which will form collagen based off stuff that we have just learned? Let me highlight this first one here. Anyone know which of those three, what sequence does this represent from those three options? Let's see, this one looks a little bit weird. 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. This must be what? Alpha keratin. There's a 7 residue repeat. This next one here, oh, there's a lot of prolines and glycines and weird looking amino acids. That must be what? The one with a lot of prolines and glycines and hydroxyprolines is collagen. Okay. And then this last one here at the bottom with the alternating pattern of glycine, alanine must therefore be beta keratin. Okay. So chapter 6, as you can see, is just a bunch of memorization stuff. Let me make sure I didn't forget anything. 1 second. Yeah, for chapter 6, that's all you need to focus on is those type of memorization things. All right, exam review is going to start at 6:30 all the way in PS 227. Unfortunately, there is already a line at the room. I actually stopped by the room, so don't fight each other or anything. Be courteous to each other. Everything's going to be recorded. All right? I'll be there in 30 minutes.
So you're still doing more right now?
No, this is it right now. This is just for the chapter 6 stuff.